Welcome back to Upregulate. Human mesenchymal stem cells, also referred as mesenchymal stromal cells or medicinal signaling cells, are the most multipotent adult stem cells in the human body and play a crucial role in regenerative medicine. These cells have the ability to self-renew, produce trophic factors, and promote the development of other mesenchymal cells. Additionally, MSCs have the ability to modulate the immune system, improve the microenvironment around damaged tissues, and aid in tissue repair. As a result, they are widely used in clinical trials and have been extensively studied to understand their applications in regenerative medicine. The fields of cell therapies and regenerative medicine are experiencing tremendous growth in biomedical research. This video aims to outline the characteristics of MSCs, their use in the treatment of inflammatory diseases, and ongoing clinical trials that are exploring the use of various types of stem cells. Amongst other important characteristics, MSCs have the ability to stably self-renew, modulate the immune system, secrete trophic factors, and selectively migrate to injured tissues. MSCs are particularly useful in allogeneic transplantation as they express low levels of HLA class I molecules and do not express HLA class II molecules, which are both essential to the graft versus host response. The mechanisms that allow MSCs to self-renew and grow in multilineages remain unclear. However, the methods of action that allow their biological and regenerative effects are mainly due to the secretion of factors that control cell growth, possess anti-inflammatory qualities, and facilitate communication between cells. MSCs modulate both the innate and adaptive immune responses through the release of many trophic factors such as cytokines, chemokines, growth factors, lipids, proteins, and even DNA material to remodel the tissue microenvironment. MSCs essentially act as the command centers that allow recruitment of all immune modulators in the peripheral blood, using each cell type to manage damage and stresses when necessary. Alright, maybe I got it wrong, maybe the MSCs aren't like Doctor Strange, they're more like the Nick Fury of the body.
I've had it with these
disease
on this
body.
It recruits the Avengers. We've got the T cells, they're like the Thor of the immune system. Then we've got like the B cells, they're like the Hulk. No. The number of MSC-based clinical trials has doubled over the last 5 years, with the number of applications ranging from cardiological disorders to treatment of COVID-19 symptoms. Heart failure continues to be a major contributor to death and disability, making the development of new treatments a primary objective. MSC-based cell therapies has shown promise as a potential treatment for both acute and chronic heart failure, by allowing the growth of new blood vessels and reducing the death of cells due to lack of blood flow, showing that this strategy has therapeutic potential. As MSCs have the ability to regulate the immune system and minimize inflammatory damage, this offers a promising new approach to treating immune-related illnesses. These properties has also allowed them to be used in COVID-19 treatment trials. Critically ill patients suffering with COVID-19 related symptoms have had the option to be treated with clinical trials such as MSC therapy, which has been shown to be effective in alleviating symptoms in the later stages of the disease. However, further studies are needed to make valid conclusions. The benefits and potential uses of MSCs for regenerative medicine have been recognised and covered extensively in literature. However, there are still several obstacles to be addressed before they can be applied clinically. Despite a decade of clinical trials indicating no significant adverse effects from MSC transplantation, the long-term risks associated with MSC therapy are not yet thoroughly investigated. Potential risks include the possibility of incorrect differentiation, suppression of the immune system, and an increase in cancer progression. Despite being deemed safe by most regulatory bodies, further investigation is necessary to fully understand the potential risks of MSC therapy. Several studies have shown that MSCs may change their intended function after being transplanted. In some cases, these cells have been observed to partially transform into unwanted cells, such as adipocytes. Other studies have revealed that MSCs can undergo chromosomal abnormalities and behave similarly to tumour cells after just a few cycles of growth in the laboratory. There are also concerns about potential risks associated with MSC transplantation, such as immunosuppression, with some evidence showing that MSCs can impair the host's defense against infections. Additionally, there is a potential risk of MSCs transforming into cancer cells, particularly when they're exposed to certain conditions, creating sarcomas. The ability of MSCs to self-renew, produce trophic factors, and promote the development of other mesenchymal cells make them a primary target for research in current and future clinical trials. However, taking advantage of MSCs and their properties for therapeutic applications will require a deeper understanding of how to control their differentiation both in the body and in the laboratory. Advances in the fields of bioengineering and biology will play a crucial role in addressing these challenges and creating optimal conditions for generating MSCs for clinical use. As a result, MSC-based therapies may become more widely adopted. This has been my video on mesenchymal stem cells. I hope you found it interesting. I've tried to use a few different sources to collate all the information for this video. If you liked the video, please like and subscribe, as it really helps the channel. All the best, and I'll see you in the next video. We've got the macrophages, which are like the Iron Man of the immune system.